Another tube for balance, okay? You put your sample at the top and you will hang it, suspend it in the rotor of the ultracentrifuge. And at a particular speed, high speed, more than 20,000 G normally. We do not calculate in G, we calculate in RPM. That is more simpler. RPM is simpler to understand. You can feel that how much time it is rotating per minute, like this. Whatever be the case, at certain RPM, when you rotate this, your molecules present within them, within this sample will get separated. It will get separated at a certain position. It will form a band here. Another some band is formed maybe here. Okay. This band is for something else. This band is for something else. Maybe this band is for mitochondria. This band is for Golgi bodies, maybe. There are several other bands which you might not see properly, but they're there. So you can take this out. Usually these tubes are made of plastics so that you can pierce a needle inside and you can take the sample out from there with the help of a syringe. You can suck that sample out. ঠিক আছে, এই স্যাম্পলটা এখানে ফুটো করে আমি টেনে নেব, তাহলে আমার সিরিঞ্জে চলে আসলো, আমি ফার্দার কাজ করতে চাই, মাইটোকন্ড্রিয়া পেয়ে গেলাম বা ফাজ পেয়ে গেলাম। এবারে ফাজ দিয়ে আমি যা করতে চাই, ফাজ ডিএনএ সেপারেশন করছিলাম, ডিএনএ আইসোলেশন করছিলাম, সেটাতে যাব, ঠিক কি না? এইটা হলো আরেকটা ওয়ে আছে। আরেকটা ওয়ে হচ্ছে এইটাকে বানানোর, যেটাতে কন্টিনিউয়াস ডেন্সিটি গ্রেডিয়েंट তৈরি হয়। মানে देयर इज ए कंटिन्यूअस डेंसिटी ग्रेडियंट, नीचे की तरफ ज़्यादा, ऊपर की तरफ कम, नीचे की तरफ ज़्यादा, ऊपर की तरफ स्लोली कम होता जाएगा। अपने इधर ज़्यादा रहेगा डेंसिटी, ऊपर की तरफ थोड़ा हल्का होता जाएगा। ठीक है, ये कैसे होगा, इसको समझाने के लिए मैं मेरे घर में पढ़ा था ये वाला, बड़वानी यूनिवर्सिटी से अशोक बाबू मुझे दिए थे, अशोक सर, AKM सर, प्रोफेसर अशोक कुमार मुखर्जी। देखो ऐसे दो वेल्स रहता है, देयर इज अ आउटलेट हियर एंड देयर इज अ नॉब हियर। दिस इज बेसिकली कनेक्टेड बिटवीन देयर इज अ ट्यूब which is connecting these two wells. So concept is this: you have put the high density solution in this, low density solution in this, and we know the nature of liquid, that it flows from higher level to lower level towards. So when you, first of all, there should be a knob also here. এখানে একটা knob থাকা দরকার, বা you can connect with a tube which will have a knob. So when you open this, liquid will start falling from this into the tube. At the same time, now you open this one also. So what will happen? The level of this will decrease. At the very beginning, the level of this will decrease. This will be a little higher. So liquid will flow from this to this. And this has a dilute liquid. This is a concentrated liquid. So automatically this liquid will get gradually diluted. তাই না? যেমন যেমন ফল করবে, হাই ডেন্সিটি লিকুইড পড়ছিল, আস্তে আস্তে এটা মিশতে থাকবে, it will become diluted. So it will form a gradient from higher density to lower density all along. এভাবে জোন তৈরি করতে হবে না, এটা গ্রেডিয়েন্ট মেকার বলে এটাকে, the gradient maker. So using this kind of, this is a comparatively simpler. You don't have to wait for Onishda. You can use this instrument rather. So in that case, you will just load your sample here, and it will again, when you run it, it will form bands at different positions that you can take it out with the help of a syringe, like that. And another one that is called isopycnic density gradient. This is also why this band forms here. Why this band forms here? Because the density of this and this matches. That's why this band forms here. This is also one type of, you can say, isopycnic density gradient. But in isopycnic, true sense, isopycnic density gradient centrifugation, the medium that you are using, usually it is cesium chloride or it is percol. You mix up the sample with the medium. The sample and the medium, they are mixed. I mean, this is the sample, homogeneous, and this is the medium. They are mixed together. This is the sample and the medium. They are mixed together. They become distributed all along at the beginning. All types of concentration get distributed homogeneously. But when you run in the centrifuge, it automatically forms a gradient. The high density molecule, they move at the bottom. The less density molecule, they gradually are on the upper side. Okay, and these substances, particles, they also get separated accordingly. They also get separated and form bands like this. So this is called isopycnic. Isopycnic means same density gradient. The band will be formed at a position where the density of the medium and the density of the particle matches. They're iso. Okay. So at the end you will get like bands like this, and then you will separate this. This is also called equilibrium density gradient centrifugation, because here there is an equilibrium between the density of the particle and density of the medium also. This example, cesium chloride density gradient, that Michelson and Stahl's experiment. You remember that? Michelson and Stahl. I don't remember Michelson spelling is S-O-N or what. Michelson and Stahl's experiment. This experiment was on what? Can you recall anything? It was there in higher secondary also, I think. DNA replication is semi-conservative in nature. DNA replication is semi-conservative in nature. That was proved by Michelson and Stahl using N15 nitrogen. Heavier nitrogen in the DNA. Do you remember this or not? Don't remember. DNA replication, this is double-stranded DNA. There were theories at the very beginning that DNA replication is conservative, and another one is it is semi-conservative. So Watson and Crick, they drew a structure like this when they were giving their theories. They drew a structure like this. That replication is going on. This type of diagram. Okay. In this diagram, it is visible that the replication is semi-conservative in nature. These are the parental strands, and these are two daughter strands. So parental strands remain as it is, and the daughter strands are copied. They make a copy of the parental strand. T against A against T, G against C, C against G, like this, depending on the complementarity. So this is called semi-conservative replication. Conservative replication will be like what? These two molecules, when they are getting replicated, they will remain as it is. They will remain as it is. The new daughter molecules will be two new daughter molecules. So this does not happen. This is not true. Does this happen? No. This kind of replication. But in 1940s, 1945, 50, that time, people did not have as many tools to prove this, that how does it occur. Okay. So they had inferred different models. Another one, that the replication occurs in a fragmented way. This molecule replicates in a fragmented way, like this. And like this. In between, new DNA are being formed, like this, discrete way. That is not also true. This was probably given by Delbruck, who was working on crossing over kind of recombination. So he thought like recombination, replication also occurs that in between the parental DNA, new daughter DNA strands are synthesized within the parental DNA. But that is not true. The parental strand remains as it is, it makes a copy of it, each of the two strands. So it is semi means half conservative, half new. Isn't it? This was proved by Michelson and Stahl by using N15. N15 is the isotope of nitrogen. Normal is N14. So why I am saying this? Because it is related with this isopycnic density gradient centrifugation. So what was the experiment? They grew the E. coli cells, E. coli culture in a medium that contained N15. Suppose N15 ammonium chloride. Use ammonium chloride which has a nitrogen with N15. So what will happen? All nitrogen containing compounds, cell মধ্যে all nitrogen containing compounds, the nitrogenous bases means the nucleotides as well as the amino acids also, they will incorporate this nitrogen within them. They grew E. coli cells for several generations within this type of medium having N15 ammonium chloride. So all nitrogen containing compounds become heavier, containing N15 nitrogen within it. Nitrogenous bases A, T, G, C, they are called nitrogenous bases because they have nitrogen within them, isn't it? So all those nitrogens those were N14 will now become N15 because they have grown for several generations within this. Now they take it out from here and transfer it to a fresh medium which has N14 ammonium chloride. So now the cells will start, the molecules will start incorporating N14 now. Instead of N15, it will start incorporating N14. So let me wipe it out first. So when you take cells from here, if you take cells from here, make DNA, separate DNA, and do an ultracentrifuge, isopycnic density gradient centrifuge, you will get a band. It will be shown like this horizontally because you are rotating horizontally. So in your books, the diagrams will be like this. You might be confused why it is like this. What is it? It is centrifuge tube actually. Okay. So here you are getting a band for the heavier DNA. Your DNA is heavier because it contains N15. N15 is heavier than N14. So it will form a band, suppose here. This is heavier band. Okay. Now, from the first generation, if you take cells from this first generation, only first generation, generation is important. First generation means what? The cells, there was a cell, it has divided into two cells. So there was a DNA that has divided into two DNA. If it is semi-conservative, it will be like this. If it is conservative, it will be like this. If it is discrete, it will be like that, the recombinant type. Okay, so we have to prove this. They are trying to prove this. Which one is right? This theory or that theory? Which one is right? By this experiment, they can prove it. You can prove it. At the first generation, what will happen? All the DNA, not all the DNA. Yeah. Here the DNA was what? N15. Here it will become N15, N14. Isn't it? N15, N14. Okay? So they will become little lighter. ठीक है? थोड़ा हल्का हो जाएगा. तो if you run a centrifuge, if you do a centrifuge here, your band will be formed. Now it will be formed here. थोड़ा सा ऊपर हो जाए because हल्का हो गया. Isn't it? একটু হালকা হয়েছে, এটাকে বলবো লাইটার, এটাকে হাইব্রিড ব্যান্ড বলবো, কারণ এটা হাইব্রিড ফর্মে আছে। এখন আমি বুঝতে পারছি না, এখন আমি লাইটার বুঝতে পারছি, ওর থেকে লাইটার হয়ে গেছি, তো এখন আমি এল লিখতে পারি, পরে এটা চেঞ্জ করব। কেন চেঞ্জ আমাকে করতে হবে? যখন আমি when you will allow the cell to grow further generation, if you take the cell from the second generation से लेने से क्या होगा? सेकेंड जनरेशन मीन्स ये दोनों একটু অগোছালোভাবে হয়ে গেছে। এইটা আবার এই দুটো এইখানে যে সেলের মধ্যে, এটা সেলের মধ্যে আছে, ঠিক না? একটা সাদা একটা ইয়েলো, সাদাটা আবার একটা ইয়েলো নিয়ে আসবে, আর ইয়েলোটা আবার একটা ইয়েলো নিয়ে আসবে, কারণ এখন কিন্তু ইয়েলো মিন্স N14, একটা স্ট্র্যান্ড অলরেডি ইয়েলো আছে, N14 ওয়ালা আছে, তো সেম মিডিয়ামে রয়েছে, তো অন্য স্ট্র্যান্ডটাও N14ই হবে, কিন্তু এই ক্ষেত্রে একটা স্ট্র্যান্ড তো সাদা মানে ওল্ডটা রয়েছে, তাই ও থাকবে, নতুন স্ট্র্যান্ডটা N14 হবে, তাহলে এটা হয়ে যাবে N15 N14, আর এটা থাকবে N14 মানে আরও লাইটার হয়ে যাবে। তাহলে তুমি সেকেন্ড জেনারেশনে যদি রান করো ডেন্সিটি গ্রেডিয়েন্ট সেন্ট্রিফিউগেশন, তাহলে তোমার এই ব্যান্ডটা থাকছে, দিস ব্যান্ড রিমেন্স, isn't it? This band remains as it is. There is another band that is formed that is now lighter, so this will be called hybrid band now. Now you tell me, if this has been true, if it had been a conservative DNA replication, then what would have happened? First generation e heavy band, light band, duto band petam, पाइनी। फर्स्ट जनरेशन में ही तुमको दो बैंड मिलता जो नहीं मिला, पहला जनरेशन में इसमें से ओल्डर मीडिया यू गॉट ए सिंगल बैंड, बट इन द नेक्स्ट राउंड ऑफ रेप्लिकेशन এই ব্যান্ডটা থেকে যেত, এটা থেকে যেত, কারণ কনজারভেটিভ বলছে, তো কনজারভেটিভ মানে কি, হেভিয়ারটা থেকে যাবে, নতুন যেটা তৈরি হবে সেটা লাইটার হবে। যেহেতু আমি এখানে কোনো ব্যান্ড পাচ্ছি না, তাহলে এই থিওরিটা ঠিক নয়, rejected, conservative model rejected, but you cannot reject at this stage that model, recombinant model বা discrete model, কি নামটা ওর ভুলে যাচ্ছি আমি। डिस्पर्सिव मॉडल, ওটাকে বলে डिस्पर्सिव, डिस्क्रीट को जो डिस्पर्सिव मॉडल, वो जो जिसमें रिकॉम्बिनेंट पार्ट है, इफ इट इज a dispersive model, then up to this you are confused, it may be dispersive model, it may be semi conservative. At first generation you can't say, you have rejected that one conservative model at this stage, but you are not able to reject the dispersive model, বিকজ ওটা হলেও এটা লাইটার হয়ে যাবে, ওল্ডারটা থাকবে না। সেকেন্ড জেনারেশনে যাব যখন, তখন ইউ ক্যান রিজেক্ট দ্য ডিসপার্সিভ মডেল, বিকজ যদি ডিসপার্সিভ হয় তাহলে ডিসপার্সিভ মডেল কি বলছে, হোয়াট দ্যাট ইজ সেইং, দ্যাট দি ওল্ডার উইদিন দি ওল্ডার লাইটার পার্টস আর বিং ক্রিয়েটেড, ইজন্ট ইট? নিউ ডিএনএ ইজ বিং ক্রিয়েটেড লাইক দিস। সো হোয়াট উইল হ্যাপেন ইন দ্য নেক্সট জেনারেশন অলসো, when this will get separated, within that the thing will happen, within that new DNA will be synthesized again, so it will become lighter and lighter without leaving any heavier DNA. Means hybridization, if you say, becoming hybrid will increase, it will get diluted rather, if you say that both the chromosomes will get diluted by the new N14 DNA. In that case, this band will shift upward, there will be no hybrid band. If dispersive model is true, then there would not be any hybrid band, but since there is a hybrid band, it will be produced only when one remains N15, another is N14. That is in the case of semi-conservative. So this proves that the replication is semi-conservative. It is not there in the syllabus right now, but it is related with this, with isopycnic density gradient centrifugation or equilibrium density gradient centrifugation. You can use this example to explain this. Huh? Sometimes what happens, in my case I used to do this. I have this idea of semi-conservative model. I have forgot about the isopycnic. I have not prepared it properly. I don't have a matter to write like that. So, but I know a little bit that, okay, isopycnic density is related with a similar density or something. So, I wrote a para about isopycnic density gradient, but it is a 6 marks, 8 marks question. Now I want to lengthen it. So, I'll give this example. For example, this my score will be at a good stage. ये कभी-कभी during your examination कभी-कभी मेकअप देने का काम करता है. हाँ, होता है ना, हर जगह आप अच्छा छक्का चौका नहीं मार सकते. कभी-कभी फंस गए तो कुछ दूसरा उपाय से बैट लग गया किसी तरह से, चलो वो भी करना पड़ता है. ठीक कि नहीं? तो ये था इसका कहानी, सेंट्रीफ्यूगेशन रिलेटेड थिंग. বোঝা গেছে? দেখো পুরো গল্পটা আমি এখানে করেছি. কটা বাজলো?